Thank you.
First of all, thank you, Professor Hefti, for joining us at the ungodly hour of 5:30 a.m. in the morning for you. It's really a pleasure to introduce you to all our members. In fact, I think if it wasn't for COVID, I would probably not have met you. Dr. Hefti graduated from the Mount Sinai Medical College and is currently faculty at the Neurosciences Division in Ottawa, University of Ottawa. I think while his passion may be for deciphering the tau pathology in developing brains, he rose to the challenge of that COVID-19 has kind of thrown at all of us, and is one of the select few neuropathology COVID warriors who has kind of braved the virus to conduct autopsies. So it will be a lesson to all of us, I think, not just to understand what the virus is doing to the brain, but to emulate his example and start doing autopsies ourselves here. I know we are all dying to listen to him. Sorry, no pun intended there. So, Professor Hefti, the floor is all yours.
Okay. Okay. Are you able to see my cursor here, the full screen?
Yes, sir, both. Thank you.
Great. Well, thank you very much for that introduction, and it's a pleasure to be here. It's as a former surgical resident before I switched to pathology, this is actually not that early, so it's not too bad. And as I said, it's a pleasure to be here. It's reassuring in these fractious and isolated times to be reminded that the diseases we see and diagnose are universal, regardless of where we're actually geographically located. So I'm going to be talking briefly about the neuropathology of COVID-19. As Dr. Mahadevan mentioned, this is an area that is very much in flux and subject to a great deal of controversy. And there is sort of a constant stream of reports, case series coming out in this area. So as Dr. Mahadevan mentioned, I'm at the University of Iowa. This is sort of the picture of Iowa that the state would like people to have. Unfortunately, for those of you who watch or follow American politics for sort of as the live version of a disaster movie, this is sort of what we've become better known for. It continues to puzzle many of us here that you in India are able to run an election with the better part of 1 billion people and have results in 2 days, and we apparently can't do anything like that for a third as many. So we may have to get some advice from your elections, folks. Anyway, the things I'm planning to cover are technical aspects of doing COVID-19 autopsies, and then sort of the paired questions of does COVID actually infect the brain, and the neuropathology of COVID-19, direct infection or otherwise, and then just sort of very briefly future directions for this field of inquiry. So when the COVID pandemic really started taking off in New York and in other parts of the US, there was a lot of sort of question about the usefulness or the safety of doing COVID autopsies. And the general reaction, I think, of a lot of pathologists here was something like this, that, you know, it was equivalent to doing an autopsy on someone who had passed away from Ebola or one of the hemorrhagic fevers. In reality, there are a number of institutions here in the US that I'm familiar with. Some of my collaborators, home institutions, have actually not permitted them to do autopsies on patients with COVID-19. I believe the Indian Council of Medical Research has been discouraging medical legal autopsies on COVID-19 patients. We actually started doing these cases relatively early on in the pandemic here in Iowa, and have been able to continue to do them safely. We've not any issues with infection of autopsy staff or pathologists. And basically, what we do is relatively simple. It's an adaptation of the protocols we use for Creutzfeldt-Jakob. But we do our autopsies in a negative pressure autopsy suite that's thoroughly cleaned after each case. The personal protective equipment we use is essentially the standard we use for any autopsies. So N95 masks, surgical scrub attire, fluid impermeable gowns, and so forth, which are all either sterilized or incinerated after each case as appropriate. The one real variation we made was that the removal of the calivarium to access the brain is done with a striker saw after placing the entire head inside a plastic bag in order to retain the aerosolized bone dust within the bag, which is then again incinerated. And again, this is an adaptation of a technique we use for Creutzfeldt-Jakob, and I imagine a lot of you do likewise. We also just make sure that we have minimal staffing in the suite. These cases initially had attracted a lot of interest, but we limited it to the resident or staff pathologist doing the case and one assistant when necessary. So we've thus far, as I said, been able to do these cases relatively uneventfully. We haven't done as many as some of the folks in New York City or Italy have, but we have been able to do them when otherwise indicated. And some of these were done specifically for the research interest to define the pathology of COVID-19. There are guidelines available. I didn't see any from any of the societies in India, but there are guidelines from the Centers for Disease Control here in the United States and the Royal College of Pathologists of the United Kingdom for carrying out autopsies in COVID-19 patients, which are essentially similar to what we do here. So my first thought when I started discussing this with some of my colleagues in forensic pathology and actually started discussing these cases with Dr. Avindra Nath at our National Institutes of Health, who I believe is a friend of Dr. Mahadevan's, is that this should be relatively straightforward. You have a virus that appears to show tropism to the olfactory mucosa, most likely penetrates the brain. You have a direct encephalitis and a certain amount of inflammatory response to that, causing damage to various structures within the brain and thus causing neurologic symptoms. Unfortunately, as you'll see, it turned out to be not anywhere near that simple as things in science and medicine often are. So the first question is, does SARS-CoV-2 actually infect the brain in humans? So this is actually work that was done by Dr. Stanley Perlman here at the University of Iowa. You'll note the date. This is 1988 on the left and 2008 on the right. So the one in 1988 was with a mouse coronavirus. Dr. Perlman has been studying these viruses for the better part of 40 years at this point. And this is in situ hybridization with 35 sulfur radioactive probes, showing at the upper right here in B, a infected mouse with high signal in, or this is, I believe, a mouse pup, a high signal in brain and spinal cord. 20 years later, after the first SARS outbreak in Hong Kong, Dr. Perlman was doing mouse experiments in human ACE2 transgenic mice with the first SARS-CoV virus and found, again, infection of the brain, as you can see here, by immunohistochemistry. I believe this is for the spike protein. Now, of course, these are mice. These are transgenic. There have been recent descriptions of the SARS-CoV-2 virus causing similar effects in these human ACE2 transgenic mice, which Dr. Perlman created some years ago. But again, they are mice, they're transgenic, and they're being infected with a human virus. There's a very interesting paper that came out recently looking at human brain organoids, which I also work with in my lab, and showing that the SARS-CoV-2 is capable of infecting organoids. So AB4 is a antibody. I don't remember which protein it's for within the virus, but it marks virally infected cells. So these are organoids grown from human embryonic or induced pluripotent stem cells. They're then transfected or exposed to virus. The virus infects them, and then there you see co-staining with viral antigen and with tunnel, showing cells that are undergoing apoptosis. The same group actually showed tau mislocalization in these same cells, which is something of my particular interest to me, given my research background, but the mechanism and sort of implications of that remain to be determined. So this is actually the first Iowa COVID autopsy we had. I used, or this is work that was done in my lab by one of my technicians and lab manager. So what we did is we took control and COVID-19 olfactory bulb, and then we used RNA in situ hybridization using this RNA scope platform with a probe to a nucleic acid sequence that's unique to the virus. This is actually commercially available from the manufacturer, ACD Bio. They very quickly rushed this probe out. And what you can see is the red is positive signal, and we saw what looks like viral nucleic acid in the olfactory bulb of the case, not control. We're working with Dr. Perlman to extend this further. And not surprisingly, other groups have looked at this as well. These are some folks in Germany, and here they're looking at human, actually the olfactory mucosa. So actually this should be mucosa, not bulb, for which I apologize. And basically what they're showing here is that different neuronal markers here are co-localizing with SARS-CoV spike protein here. So it seems very likely that there is viral involvement of the olfactory mucosa and the olfactory bulb, although obviously the implications of that are not entirely clear. Would potentially explain this sort of anosmia that is often seen in these patients, however. The issue becomes that when you start asking, okay, so does the virus actually infect the brain parenchyma? And if you actually start looking into the data, a lot of it becomes a little difficult to interpret. So most of the data that's out there are things like this. This is from two groups in Germany doing, again, immunohistochemistry. I believe this was the spike protein again. And the issue is that you can see positive staining, but for all of us who spend significant portions of our lives looking down the microscope at brown stains on blue, it's not the most impressive. You have a few positive blotches in endothelial cells here and here. You have this sort of dry wash here in a nerve root, and then a few positive clusters here. And this does seem to be real, does seem to be positive, although with the caveat, as one of my teachers in residency pointed out, you can make anything positive if you leave the antibody on long enough. So there does appear to be some infection of the brain, the central nervous system, away from the olfactory bulb by virus, but the amount seems to be small, and some of the data is a little unclear. Interestingly, if you look at the Allen Brain Atlas human single-cell transcriptomic data, so the right side is a scatter plot with using principal component analysis of single-cell, publicly available single-cell transcriptomic data set. And if you look at the right side is cell types, so colors are different cell types, and the left side is ACE2 expression. And while there are scattered cells in the neuronal populations, for example, that show, so you can see red dots mixed in here, some expression of ACE2, by the overall level is low, and the percentage of cells expressing it is actually quite low. And there are a number of groups who have done PCR and sequencing studies, including Dr. Nath at NINDS, and the results of that have been somewhat ambiguous. And I'll summarize the existing data at the end of this next portion of the talk. But basically, a subset of patients do seem to have low-level PCR or IHC detectable virus in the brain parenchyma. Not all do. The majority appear not to, and the levels overall seem to be quite low. So again, the significance of that remains unclear. So does SARS-CoV-2 infect the brain? Definitely in mice, transgenic mice, definitely in organoids. In living humans, perhaps, most likely, yes, in the case of the olfactory bulb. Most likely, yes, at least in some cases in the central nervous system. The data correlating that with the neurologic findings remains sort of an open question. And it's probably, in most cases, focal and relatively at low levels. Which, again, the significance of that and how that correlates with symptoms and neurologic symptoms remains unclear. So regardless of whether you actually have virus, does COVID-19, so the disease, the syndrome, affect the brain? Which is actually a separate question. It's one that's provoked a lot of interest, both in the scientific and the popular literature, sort of an example of various headlines from different popular media in the US, the UK, and India. And essentially, there is this phenotype of people who have cognitive dysfunction after recovering from COVID-19. Unfortunately, as some of these articles have pointed out, there's a major confounder that these folks are all critically ill. Not all, but most are critically ill. They're in the ICU. They're under a lot of stress. So there's a lot of potential confounding factors for cognitive dysfunction. So again, one of the first things we did was just to look at microglial activation in the olfactory bulb in some of our first cases. Here's an example on the right. So you have, this is IBA1 immunohistochemistry, and you see, actually, sorry, not IBA1, CD68. And you can see that there is an increase in microglial, sort of in positive staining. It looked like reactive or activated microglia when you look in the olfactory bulb. Because of, we have some particular research interests here, Dr. Richardson, in respiratory regulation. When you look at we looked at the medulla as well and saw similar findings that in the COVID patients, you see very clear microglial activation. And reassuringly, this is something that has been seen by other groups as well. So this is a large cohort coming out of Germany, I believe University of Heidelberg, and they also noted in multiple brain regions, both gliosis, as indicated by GFAP in the left column, and HLA-DR indicating microglial activation in the center. And they also identified increased numbers of perivascular lymphocytes in a number of their cases, as shown by CD8 staining in the right-hand column. We have also seen this sort of perivascular lymphocytic infiltrates in our cases, and they've been described by several other groups. This is work that was done by our lab in collaboration with, where we provided a lot of the specimens in collaboration with, Dr. Nath and several groups at the NIH, who did high-resolution ex vivo MRI on a high field magnet on tissue from, this is actually that same case I showed you the RNA scope from at the beginning. And what's interesting here is, so this is a section of pons, and you can see that there is a signal abnormality here in the central pons, which when is not readily apparent on H&E, but when you stain with fibrinogen, it shows evidence of leakage and a leaky blood-brain barrier in these same cases, in the same tissue. So this is actually the same tissue that was MRI'd here, was embedded and cut and stained on the right. Now, this is a sort of summary figure from the largest German study. And the reason I'm showing this is the different colors indicate different degrees of gliosis, microgliosis, astrogliosis, T lymphocyte infiltrates up here. And you can see this looks like a drawing that was made by my children with crayons. So the point here is there's a huge amount of heterogeneity in the neuropathology of neuropathologic findings in this disease. And the bottom here, this was multiple ways of detecting the presence of SARS-CoV-2. And you can see, so purple is positive, green is negative, and gray is not assessed. So again, this highly variable as to whether you can detect actual virus in the brain, and often it is at very low level detection by PCR. So these are the published case series that, based on a search I did yesterday to make sure I caught all of them. There are, the largest series is here, 43 patients by Matschke et al. In Germany. We have 18 patients. This is currently in press by Dr. Nath and a group of collaborators, including my lab. And basically the common themes here are that the findings are often subtle. A lot of groups have observed microglial activation, perivascular lymphocytic infiltrates. A few cases of encephalitis have been seen, especially brainstem encephalitis. But again, this is rare. To no one's great surprise, a lot of these cases have acute hypoxic ischemic injury. The other thing that has come out of this data was initially first described in systemic organs, but is a procoagulatory phenotype, where these patients are prone to have microinfarcts and have intravascular thrombi in the brain. So the sort of a somewhat pleomorphic pattern of neuropathology, with the overall theme being vascular perivascular inflammation, microglial activation, and in some patients, rare patients, encephalitis, and in some patients, this sort of procoagulant stroke-like phenotype. So what this means is that, as often is the case in medicine, things are probably a lot more complicated than we initially thought. So it's fairly well established, or it seems to be fairly well established, that this virus does have a certain tropism for the olfactory mucosa. It is capable of accessing the olfactory bulb to some degree. You may have some low-level viral infection of the brain in a subset of patients. The exact frequency of this is unclear, and it is relatively clear that you have a inflammatory response within the brain in some patients, and that constitutes or is composed of some perivascular inflammatory lesions. In some cases, this is more lymphocytic. In other cases, you start seeing macrophage aggregates and even demyelination, perivascular demyelination. One ADEM-like syndrome has been described. And then thrombosis. So some of these patients, again, do have multiple infarcts. And the virus does seem to have a procoagulatory phenotype in some patients. Now, what complicates this is that, as I said, these patients are all critically ill. They all have systemic viral infection, so which in, again, a subset of patients who become very ill with this virus triggers systemic inflammatory response, cytokine release, and so forth. And the problem is it's not clear to what degree the responses we see in the brain are due to direct infection or due to a systemic inflammatory response that secondarily affects the brain. And there's even data to suggest there may be an autoimmune component to this with cross-reactivity of antiviral antibodies to normal human proteins, causing an autoimmune component. The other thing that hasn't really been, that most of these studies are missing, is controls from patients who are critically ill. Because again, as we noted, these patients are critically ill. There's a known role for critical illness in causing cognitive dysfunction. And it's also not clear how many of these findings we're seeing are truly because of COVID infection, either directly or indirectly, and how many of them are evidence of systemic critical illness. Again, we're sort of used to what is and isn't normal in critically ill patients, but at the same time, that's something that I think in the future does need to be addressed and accounted for in these studies. So there's actually a group here in Iowa that's starting to do long-term cognitive follow-up on patients who survive COVID-19 to try to better understand these cognitive phenotypes. And we're hopefully going to have the ability to recruit some of these patients for brain donation as well, so we have pathological correlation for their cognitive findings when they pass away, hopefully of non-COVID related causes. So does COVID-19, the syndrome, affect the brain? Yes, it appears to in a significant fraction of patients. The most commonly described findings are vascular and perivascular inflammation, microinfarcts, thrombotic events, and leaky blood-brain barrier. As we said, there are poor to no controls in a lot of these studies to account for critical illness. There's poor control for the duration and severity of illness, in part because it is, as you all know, difficult to get these patients who pass away to autopsy. And as you've noticed, most of the data comes from Western Europe and the United States. So we have very little data on the neuropathology of COVID from outside those areas, which also limits our ability to generalize these findings to the vast majority of the population. So with that, I will wind up. I just wanted to acknowledge our collaborators who were involved in that study that's currently in press, Dr. Avindra Nath at the NIH, Dr. Folkert, who's Chief of Neuropathology at the New York City Medical Examiner, Dr. Pearl, and then my colleagues here in Iowa. And thank you all for listening to me.
No. Hello, Hefti.
Can you hear me?
Am I audible?
You are.
Yes, ma'am. Yes, ma'am.
Yeah. Dr. Hefti, it is such an excellent lecture on the neuropathology of COVID, and we are all wondering what is actually happening, and you have beautifully illustrated the various aspects of this. I have one question. It is from the olfactory mucosa that it goes to the brain. What is the predominant portal of entry? Is it a perineural invasion or through the blood brain or due to endothelitis or due to epithelium to the brain? How is the actual portal of entry into the brain?
So that's a very interesting question. And unfortunately, the answer is that I don't think we can really say for sure. I think we can say that there is, in some patients, infection of the olfactory mucosa and olfactory bulb. So I think it's fairly clear that that is a possible route of entry. But again, the problem is that we don't have enough data, I think, to conclude that, to be able to correlate that with the presence of virus in the brain per se. So it's quite possible that in some patients the portal of entry is olfactory, and in others it's a hematogenous spread. I think we just don't know. I mean, speculatively, I think it makes the most sense for the portal of entry to be olfactory, just because that sort of mechanistically seems to be it's the way you get you contract the virus. It does have a tropism for the respiratory mucosa and for the olfactory mucosa. So I think one could speculate that that is in fact, and it would make sense for that to be the primary route of infection. But again, I don't think we can be sure. There's just not enough data at this point in time to make any definite conclusion.
Thank you very much, Dr. Hefti.
Thank you.
Any more questions? Dr. Anita, can you read those questions?
Yeah, thank you, Professor Hefti. Very, very informative talk. I think it has stimulated the audience. There are quite a few questions, but I'll pick a few. There is a question on whether there are any age-related differences in COVID-19 neuropathology.
So that's also a very interesting question, and unfortunately, I think a lot of my answers are going to be this general strain if we don't know. Most of the patients that have come to autopsy, both here and in most of these series, are older. So we have not had. We have had one case of a fetal death that we suspect may be due to maternal COVID infection, where there was actually virus detectable in the placenta. We have ourselves not had any pediatric deaths. I have colleagues at the Children's Hospital of Philadelphia who have had pediatric deaths, but unfortunately, for logistic reasons, they were, in a lot of those cases, not able to secure pathologic examination of the brain. So I think we just don't know. And one of the problems is, as you all know, some of the findings that have been described, perivascular scattered lymphocytes, a few perivascular macrophages, you can see that in an older patient with hypertension, with chronic hypertension. So some of this stuff becomes a question of, are we seeing, and I have seen people get all excited about, some of our residents is like, yes, we have some perivascular macrophages, but this is an 80-year-old with chronic hypertension, so you can't really hang your hat much on that. Whereas obviously, if you have some of these cases where you have like significant lesions, then you can say for sure that that's not normal. But I think that remains, again, something to be further analyzed. And I think this is also a question that will be helped as more of these long-term studies come online. And, as potentially, for example, with Dr. Mahdevan's brain bank, you start having donors who are recovered from COVID-19, no longer have virus, and are maybe dying a year or two later. I think it will be very interesting to see how these findings develop with time.
Sure, Dr. Hefti, I'm really dying to do autopsies, but I do understand that ICMR is now going to be lifting the ban, thanks to everyone putting pressure on them. Essentially, other questions in the chat box are what you have mentioned in your talk. Many of them want to know how specific are these findings really for COVID-19? Why can't it just be part of a viremia? Why can't it just be immune response, maybe sepsis, which I think you've covered very well. There is one question which I think you would like to take. Is there any propensity to get fungal or parasitic infection in patients with COVID-19? I think several of our clinicians have also come back to us and said that many have had zygomycosis after recovery from COVID. So what are your thoughts on that?
So that's also very interesting. I mean, I actually have not I have not seen that here in Iowa. I have not heard. I haven't heard that from my colleagues in New York City who have done a number of these autopsies. So it's actually that's actually very interesting, and I don't know whether that represents. I don't know whether that represents different patient populations or whether that represents. I don't know. It's interesting because it's not something that I have seen here I've seen here in Iowa. So I'm really not sure in terms of secondary infections. Yeah, I mean, it would make sense for it would make sense for these folks to be at higher risk, but it's not something I've really seen. Dr. Challa, you're muted.
So I think a lot of things will continue to remain conjunctural. You kind of passed the buck back to us, telling us that we should start doing autopsies, I think. So on that note,
one comment.
Yes.
Mucormycosis is also known to spread by perineural invasion from the nasal mucosa to the brain. Whether there is any connection between these two things, that is why the incidence of mucor after COVID increases, is one thought. Another one is aspergillosis in the lung is reported after COVID-19 infection. A few reports of aspergillus were reported after COVID. So I think because these are rare and these are few cases, maybe the evolving picture has still to come.
Yeah. So there's actually one. Sorry, one of the cases, I believe, in the German Matschke paper did have mucor. Now that I think about it, but yeah, no, it's the disease. As several of my colleagues here on the clinical side have said, it's a very interesting disease. It's a very complex disease, and it's one we just don't fully understand yet, and probably won't for some time.
I'm afraid we'll have to move on to the next session, although I think it has really excited everyone. There are several more questions. I would encourage all of you to probably mail Dr. Hefti, and I'm sure he would be able to answer your queries. Thank you, Dr. Hefti.
Absolutely. Thank you.
It's a real pleasure having you. We move on now to the next session everyone is waiting for,